Hello everyone, welcome to this online presentation. My name is Charlotte Könders. I'm a business development manager at BioGenes, and today I'm going to present you BioGenes strategies for a successful HCP monitoring. This includes our sophisticated ELISA development strategy, supported by orthogonal methods for reagent characterization. Let me briefly introduce BioGenes as a company for those who are not familiar with our services. We support the development, testing, and quality control of new biologics and biosimilars. And apart from our kit production and analytical services, we produce antibodies. And one of our core services is the ELISA development. In this context, we offer robust and reproducible ELISA-based HCP monitoring. And today I'm going to discuss the important steps for our customized HCP ELISA development. First of all, what are HCPs? Host cell proteins are defined as process-related impurities in drug products. They carry clinical safety risk. For example, they may elicit an immune response or act as an adjuvant, and further, they may influence the drug quality by drug degradation. And the EMA and FDA, as the two major regulatory bodies, have released a set of guidelines that are constantly updated and that ensures high levels of patient safety during the clinical trials. In general, the market authorization requires a profound overall assessment of potential high risk and benefits. Therefore, an accurate detection of HCP impurities is necessary. And the enzyme-linked immunosorbent assay, short ELISA, is still considered the gold standard for HCP measurement, having advantages like high sensitivity, speed, and high throughput. Here at BioGenes, there are two options that are generally considered for HCP monitoring. This is the generic HCP ELISA and the process-specific HCP ELISA. The generic HCP ELISA makes use of a broadly acting antibody coverage approach, which is specific only for the selected cell line, whereas the customized or process-specific HCP ELISA is based on greater specificity towards the manufacturing and the processing of the particular biopharmaceutical. In the next slides, I will lead you through the steps of our customized ELISA development, which includes 3 developmental phases: the HCP antigen preparation phase, followed by the antibody generation phase, and the last one is the assay setup and qualification phase. I will start with the first one. It's the antigen preparation phase. This includes mock preparation and its characterization, and the HCP antigen fractionation. The mock fermentation mimics the production process, and the resulting mock material should represent the HCP impurities of the production process as far as possible. To achieve a good similarity of the HCP antigen preparation, the following recommendations should be taken into account for mock production. First of all, we don't have any drug substance in our mock, so use the production strain with an empty vector without the target gene. We also recommend to use same cultivation conditions, for example, medium, temperature, or viability at harvest. We also recommend to use the relevant HCP antigen fraction, for example, supernatant or lysate. And we know from prior experience that it's also really important to use an early downstream process sample as mock antigen, which then can provide a comprehensive protein pattern with almost all HCPs that are potentially present in the product harvest. There we have to keep in mind that the purification of the mock HCP may also result in the loss of important hitchhiker HCPs, which interact with our product. And finally, it is recommended to characterize the HCP antigen comprehensively with different analytical methods, for example, total protein assays or 1D or 2D electrophoresis. To evaluate whether the mock materials successfully represent the HCP composition of the product containing harvest sample, comparative 2D Dutch analysis can be performed. For this purpose, the two samples are fluorescently labeled and subjected to the 2D SDS page, and an internal standard is also included, which is labeled by another fluorescent dye and allow for the reliable overlaying of the two gels to compare the two samples. And here in the pictures you can see on the left the mock, and in the right, the total protein spot pattern of the harvest. And you can see that these images clearly show a similarity, in this case to 96%. So the mock sample represents the majority of the HCPs visualized in the harvest sample. Thus, we can recap that the mock sample appears well suited to represent the HCP composition of the harvest sample. The antigen preparation phase includes also the fractionation of HCPs. Because low molecular weight proteins come along with additional analytical challenges, they tend to be less immunogenic, so that low molecular weight specific antibodies might be underrepresented in our mixture. So our counter strategy is the fractionation of the HCPs prior to the immunization. And this can be realized, for example, by ultrafiltration or size-exclusive chromatography, and can significantly improve the antibody immune response in the end. After having prepared and sufficiently characterized the HCP antigen, the antibody generation phase is studied, which includes animal immunization, antiserum characterization, and antibody purification. As I already told you, we have now two samples, the untreated total HCPs and the fractionized low molecular weight HCPs. And we're using these two samples to immunize animals. BioGenes prefers a dual animal approach, routinely with goats and rabbits. This increases in general the probability of detecting a greater range of HCPs. Furthermore, multiple animals per species are immunized over several months to allow for both the generation of a broad antibody spectrum and the development of a robust immune response, even towards the low immunogenic antigens. And the immunization period is then continuously monitored by ELISA titer determination and Western blotting. An example of the antiserum immunoblot is now shown on the right. And then a small-scale antiserum test pool is affinity purified using the immobilized antigen material. The resulting purified capture antibody is characterized by Western blot. And for the development of a sandwich ELISA method, the purified antibody is also used as detector antibody, but then labeled with biotin. And the results are taken into account for decision making on the preferred animal species and the purification strategy. The assay performance depends also greatly on the antibody performance criteria. The antibody performance is assessed based on different quality attributes. As you can see here, on the one hand, sensitive HCP detection is required, as shown on the left. And there, typically, the LOQ levels should be below or at 5 nanogram per ml. As presented in the middle, suitable HCP-specific antibodies should also adequately reflect the log reduction of the HCP content over the consecutive downstream steps, as indicated here by the green line. And a rather poor reflection of the expected log reduction is displayed by the red line. A further important criterion is the dilution linearity, displayed here in the last graph. The red points again indicate the dilution range that does not properly reflect the expected twofold decrease over doubling dilutions. This is often due to a limitation of antibodies specific for a certain individual sample HCP. But in contrast, within the green point region, adequate dilution linearity is reached, and their maximum CVs of about 20% calculated over sample dilutions are generally considered acceptable. And of course, it has to be demonstrated that the HCP-specific antibodies do not cross-react with the product, which would occur then false positive ELISA signals. And from our experience, a sandwich ELISA setup using purified antibodies is the best option to detect a potential cross-reactivity, while Western blot analysis or ELISA titer determination of purified antibodies or crude antisera often reveal false positive signals. If all antibody performance criteria are sufficiently fulfilled, the preferred animal species is used for the second immunization round. This means that the antiserum collection is continued with the chosen animal species and upscaled for bulk production. Thereby, large antiserum volumes of equal to or more than 1 liter are purified to avoid a frequent reagent supply, and also the corresponding biotin conjugated detector antibody is prepared in large scale. and eventually suitable working aliquots of the HCP antibody reagents are prepared for long-term storage at deep frozen conditions. The following slides will lead you through the last phase. It's the assay setup and qualification phase. This includes ELISA optimization, stability testing, and qualification, also known as pre-validation. And afterwards follows the HCP antibody coverage determination and the HCP identification. I will start with the ELISA optimization. Based on the reagents prepared, the HCP assay has to be optimized with regard to reagent concentrations and incubation times. This in general ensures a higher assay sensitivity. Besides, the assay accuracy, precision, and dilution linearity has to be clarified to provide a reliable HCP determination. Most importantly, a relevant downstream process sample, for example the bulk drug substance, has to be included during the developmental steps. So if a downstream process sample is analyzed in parallel to the HCP standard, biased HCP determination can be prevented. Or it would become obvious, then we can choose the optimal assay conditions and then we can prevent it. So on the left, a typical HCP ELISA standard curve, which was generated during assay development, is shown. At last, BioGenes clients can choose then between two options: setting up the HCP ELISA as a customized ready-to-use kit, or providing just the optimized ELISA method and the critical reagents like antibodies, HCP standard, and assay control sample. After assay optimization, a system suitability test is implemented, including comprehensive acceptance criteria for standard curve characteristics, unknown samples, and an independent assay control sample. And such an independent assay control sample is analyzed in multiple ELISA runs during the ELISA development, and the target range is then typically defined as the free SD range, which is shown here on the right. And such an independent assay control sample is really an essential part of the assay setup at BioGenes. Finally, after having developed and well characterized the HCP ELISA method, reagent stability testing and qualification work is carried out. This includes freeze-thaw testing of critical reagents, such like antibodies, HCP standard, and assay control. And in case of providing the ELISA method in a kit format, real-time stability testing is conducted. Furthermore, qualification, also called pre-validation of the assays, performed according to the established guidelines and by setting acceptance criteria, which are predefined in a protocol in close cooperation with the customer. And robustness testing of the method towards varying incubation times, temperatures, and equipment is frequently included during this project stage. The last step is the reagent characterization, also known as HCP coverage. The HCP coverage analysis is required to ensure a reliable impurity measurement in the corresponding HCP ELISA method. And BioGenes currently recommends several approaches. This is the 2D Western blot, immunoaffinity chromatography, short EAC, and subsequent 2D Deutsch, and EAC in combination with mass spectrometry, short MS. I will start with the 2D Western blot. I would say it's the broadly accepted standard method for coverage analysis. Thereby, the sample of interest will be subjected to the 2D electrophoresis. Then the proteins are separated by their isoelectric point and their molecular weight. Then they are transferred onto a membrane and incubated with the anti-HCP antibody. And in the end, overlapping signals from proteins and immunostaining then can be considered as successfully covered. This technique has clearly its strengths, so it focuses on direct interactions. It is pretty sensitive, thereby requiring comparatively little material. It does not require physiological buffer conditions for a given sample, thereby enabling coverage analysis for also special sample types, such as a solubilized inclusion body. And it is a visual method, thereby allowing to look at the overall representation of a sample and the antibody performance over the quadrants of a 2D gel. However, as any analytical method, also the 2D Western blot comes along with several disadvantages. It is a denaturing method, especially the low molecular weight proteins suffer from denaturing assay conditions and low detection rates. And the dynamic range of the method is further restricted, so that protein detection may be missed due to the low expression. And having this in mind, BioGenes has meanwhile established immunoaffinity chromatography in combination with 2D Deutsch as an orthogonal method for coverage analysis. Thereby, the HCP antibody is immobilized on a column matrix and the HCP sample of interest is applied. The non-binding HCPs will end up in the flow-through, while those HCPs which are successfully covered will be captured on the column. And then, after stringent wash steps, the bound HCPs are eluted and compared with the original input sample by 2D Deutsch. And finally, a percentage coverage will be determined. IAC comes along with additional analytical strengths. So the method allows for protein-antibody interaction under nearly native assay conditions, very similar to the antibody's final destiny, which is of course the ELISA. Further, IAC has a very broad dynamic range, since it allows for the enrichment of proteins on the capture column, which helps to overcome the detection limits of low abundant and or less immunogenic proteins. And the readout by 2D Deutsch also provides visual informations. A limitation of the standard EAC is that it requires a high amount of antibodies. Therefore, BioGenes optimized the method and offers now a small-scale EAC, which permits then an 80% reduction of antibody material. However, a concern of IAC is the co-detection of indirect binders. The last orthogonal method, which is just evolving as another available tool for coverage analysis, is mass spectrometry. BioGenes offers in collaboration with AlphaLyse MS services upon request. First of all, MS allows for identification of specific HCPs and it further enables relative quantification. However, it is no visual readout, and for the evaluation, a suitable protein database is required. So let me come to an overall conclusion. Every analytical method for coverage determination has its pros and cons, and their orthogonal or multiple use provides a solid HCP surveillance strategy, including quantification and identification of potentially high-risk proteins. Now, in the end, I just want to highlight again BioGenes features and share some take-home messages with you. First, choose the right mock material. The mock material should represent the HCP impurities of the production process as far as possible. Second, the suitability of the HCP ELISA depends on the recognition of the relevant HCPs by antibodies. For this purpose, we offer a broad immunization approach, including 2 species and long-term immunization. Moreover, we comply with antibody performance criteria and we provide test purification and a comprehensive ELISA optimization. And as I told you in the end, due to the methodological limitations, the usage of orthogonal methods is the best way to assess the HCP-specific antibody coverage. Now, in the end, I can just say that we would be really happy to support your HCP ELISA project and work with you together. With that, I would like to end my talk, but not without saying thanks to a lot of people at different departments at BioGenes and especially to you for your attention. If you have any questions, feel free to ask and write us an email to service@biogenes.de or visit our website.